In this lecture, I'm going to talk about neutrophils. Neutrophils are granular cells originate from myeloid progenitor that originate from multipotent stem cells. And they are abundant cell in the body, and they are around 40 to 70% of the total white cells in the body. Neutrophils are the very cells that to arrive site of infection, and they are attracted or recruited by damage associated molecular pattern and pathogen associated molecular pattern. And as can be seen here, this is the blood vessel during inflammation. And as you know that the three major events that take place during inflammation are increased capillary permeability, vasodilation, and influx of cells. So these are the neutrophils attracted to the site of damage or site of infection by damage associated molecular pattern and pathogen associated molecular pattern, and their migration to the site of infection starts by margination. So they bind some adhesion molecules upregulated due to inflammation and due to infection. This is followed by the rolling from one endothelial to the other, then followed by the extra vasation or the third step of their migration to the site of the infection, where they reach and start to fight with the microorganism. However, neutrophils, they have the capacity to destroy and to kill pathogens by following three ways. The first is phagocytosis, which it could be complement mediated phagocytosis, as neutrophils express on their surface complement receptor number 1 and number 3, and as can be seen here, this is a bacteria when it is surrounded by opsinin C3b, so this will bind to complement receptor on the surface of neutrophils. And also phagocytosis, it could be antibody mediated phagocytosis, as neutrophils express on their surface Fc gamma R, Fc gamma receptors. These are specific for IgG antibodies. So when IgG antibodies capture the microorganism, it will bind to this receptor, and this will assist the process of phagocytosis. The second way by which neutrophils can destroy the pathogen is through expression or liberation of these granules, which include platelet activating factor, reactive oxygen species, proteases, elastase, myeloperoxidase, and proteinase 3, defensin, and matrix metalloproteinase. So these mediators can destroy the microorganisms and pathogens by different mechanisms. The third way of killing the pathogens by the neutrophils is through extracellular traps, and as can be seen here, neutrophils, they modify the chromatin, and then they produce some fibers or filaments These are known as extracellular traps. They are very sticky and they may mediate adherence of neutrophils to the endothelial cells. However, these traps or fibers, uh, they are associated with some enzymes, for example, myeloperoxidase and proteinase 3, and these are actually, they are hidden antigens and they are cryptic antigens, and so they should not be seen by the immune system, but due to expression of this, due to the modification of the chromatin and expression of these fibers, this will come out. However, when the immune response is done, as can be seen here, these pathogens are captured by the neutrophils. So neutrophil now they can phagocytose this molecule, but however the phagocytosis could be incomplete and remnants of these mediators or enzymes may stay outside in the circulation, and this will be seen by the immune system, and this will lead to autoimmune uh, disorders. And this process actually, these fibers, they are known as neutrophil extracellular trap, and the process of modification and expression of these fibers is known as